All right, so one more lab for reproduction for male and female. And so you've seen the models of the male and female reproductive system, and we've talked about the gonads, the testes, and the ovaries of the male and female. And so we need to do a little bit of histology. So there will be a couple of questions on your lab exam about the histology of the testis and the ovary, and that'll be all the histology you have. It's really not a lot compared to some histology you've done in the past. All right, so some brief histology. Most of this you've probably seen a little bit in your lecture material as well. You've got similar pictures in your textbook for reference too. So if you think back to the male reproductive system, you've got the testis, and it's made up of those seminiferous tubules. So it's kind of like that. And so you have those little seminiferous tubules, and so this is a cross section of a testis, and so you're kind of looking down into the tube. So each one of these, you're looking into the tube of the seminiferous tubule. And you'll remember that that's where the sperm are going to develop. So they're going to go from being a spermatogonia all the way to a sperm, and then out into the epididymis to be stored and then ready for ejaculation. So each one of these is going to be a seminiferous tubule. Let's see if I can, whoops. Hang on, let me make it a little bit darker or wider. Better, sort of. All right, so each one of these things is a seminiferous tubule. And so the way the sperm develop, you'll remember that you have your primordial or your spermatogonia cells. They're going to be around the outside, and as they mature, they move toward the inside of the seminiferous tubule. So what do you call the inside of any tube? The lumen. All right, so that would be something that I could say, what's the inside of this tube called? That would be the lumen. And remember, you've also got the testes. Not only do they make sperm, they also make some hormone, right? Some testosterone. And so these cells that are found between the seminiferous tubule, they're called interstitial cells. That's what these are, okay? So this is where the testosterone is made. So this will be some questions I could ask from this slide. You can zoom a little bit bigger and see a little more detail. Again, you can see the interstitial cells. You can see each one of these circles is a seminiferous tubule. The outermost ring in the seminiferous tubules are the spermatogonia. Okay, so each one of those little purple dots is the nucleus of a spermatogonia. And as they develop and they move toward the lumen, they go through their primary and secondary stages. Now, unless you're an actual histologist, I don't really expect you guys to be able to differentiate between a primary and secondary spermatocyte on this exam. So really what I want you guys to be able to differentiate are the spermatogonia and the mature sperm. And they're going to be the things that are kind of free-forming or free out in the lumen. So spermatogonia and mature sperm, you should be able to recognize. Interstitial cells, you should recognize, and you should recognize that a seminiferous tubule. If you see another circle that has red stuff in it, that's a blood vessel. Okay, so that's not a seminiferous tubule. Here we've blown it up a little bit more, so you can see the edge of the seminiferous tubule. Again, those outermost cells are going to be the spermatogonia. You can also see the nuclei of the Sertoli cells or sustentacular cells, but again, you've got to have a good eye for that, so I'm not going to make you identify the sustentacular cells on the histology exam, but you do know what they're for, right? They're kind of those nurse cells in the testis. It helps kind of guide the development of the sperm through its maturation process. Again, you should be able to identify the spermatogonia around the outside and the sperm on the inside. So here is, let's see, here's one right here, and you kind of see these, anything that has a little whip-like tail, basically all of these things in there are going to be sperm. See a little kind of a comma-shaped, spiky-looking head, that's the head of the sperm. So you should be able to identify those. Again, I'm not going to ask you to identify primary and secondary spermatocytes. So that's all there is on the male. There's no histology of the penis or anything like that, just of the testis. So the other histology you've got to learn is just going to be the ovary. We're not going to do any of the breast or of the uterus or any of the pregnancy models. So the only histology is just going to be the ovary and the testis. So this is a cross-section looking at an ovary. And so if you see these tiny little dots, these little circles, these are the primordial follicles. And if you have a microscope slide, they're usually going to be kind of around the edge of the ovary. They're just kind of big clusters. And so they're pretty easy to see. So the next thing you want to see is as they're maturing, they start to form that follicle around them. So remember, we have primary follicles and secondary follicles, and then you have a mature follicle that's going to be what ovulates your egg. And so what you're starting to look for is these oocytes that are inside of these follicles. And so this particular one right here that's surrounded by little red dashes, that's a primary follicle with a primary oocyte inside of it. Now the way I know it's a primary follicle is it has one layer of cells around it. So just one layer of cells around the egg or the oocyte tells me that it's a primary follicle. All right, so that was pretty easy. If it's a secondary follicle, let me go back to this slide. If it's a secondary follicle, you will see the oocyte in the middle. I'm going to do it right here. And then you'll see two layers of cells around it. So primary has one layer, secondary has two. Okay. So here is another example of a secondary follicle that's already started to form that antrum. So you have your secondary oocyte, that's this circle in the middle. The secondary follicle is going to be this whole structure, okay? And you can tell it's sort of got two layers of cells here, not as clear cut on this particular slide, but you know it's a secondary follicle because it has that space in it. So if it has that fluid-filled space, which is all of this stuff, we know that's a secondary follicle. So antrum is a thing I can ask you. I can ask you what kind of egg it is. It's a secondary oocyte. I can say what kind of follicle. It's a secondary follicle. I know that because it's got that big fluid-filled antrum in it. And I also know what kind of cells make up the follicle that kind of fill in this area right here. And those are granulosa cells. So those are four things I could ask you from a slide that looks like this. If you see something that's got red stuff in it again, that's kind of a bright red color, those are blood vessels, so don't worry about that. All right, so this is another secondary follicle. It's got a little more detail, a little more questions I can ask you again. So again, you've got the granulosa cells that kind of make the follicle itself. You've got the antrum, the fluid-filled space here. You have, again, the secondary oocyte. So that's this thin pink ring. So all of this inside, that is the secondary oocyte. The thing in the very, very middle is the nucleus. And if you remember, when you have that secondary follicle in the secondary oocyte, that secondary oocyte is out on a stalk. So this is the stalk, okay? And it's surrounded kind of like by a shell. So the outermost layer of the shell is called the corona radiata. And between the corona radiata and the actual oocyte is a zona pellucida. That's this white area, so clear zone. So there's 1, 2, 3, 4, 5, 6 things I could ask you about this particular slide as well. So lots of details on the secondary follicle. And that's it. So not a whole lot of histology. I know you're breathing a sigh of relief, so not too bad. All right, so I'll see you in another video. Thank you.